Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello students, are you okay? I hope you are fine. Welcome to biology lesson. This is senior 5 class. We are still on unit 15, which is immunosystem, vaccination and antibiotics. That's the name of this unit 15. The lesson of today, it is about antibodies and their function. So you have to be ready and then get your notebook where you can write some explanation. So I hope you enjoy the lesson. So what is the objective of this lesson? At the end of this lesson, you will be able to describe the structure of antibody. At the end of this lesson, you will be able to describe the structure of antibody. And then also you will be able to explain the function of antibodies. What's the role of antibody? This is what we are going to see, and make sure at the end of this lesson, you should do this. You should be able to carry out this, to describe the structure of antibody and describe the function of antibody. When we talk about antibody, what's that? Antibody is, first of all, they are protein in nature. They are proteins. They consist of a protein known as immunoglobulin. So this one, they are there just to do what? To help our body to fight against what? The germs, pathogens. So they are going to be stimulated when there is what? When there is foreign material that can harm our body. If those foreign materials that can harm our body are entered into our body, so those antibodies, they have to be stimulated in order to do what? To fight against those pathogens. Remember, pathogens, those are organisms that can cause diseases. Among pathogens, we do have bacteria, we do have also virus, we can even have fungi. All those, they can cause diseases in our body. So that's why antibody, they have to be stimulated once there is presence of what? Of those germs, of those pathogens. So those pathogens, they do have what you call antigens. So these antigens, they are the one that can stimulate the production of this antibody to work. So antibody is a molecule that's synthesized by an organism in response to the presence of foreign substances known as antigens. Each antibody is a protein molecule. It has protein molecule called immunoglobulin. Actually, they are protein. Then they are called immunoglobulin. So they are going to be synthesized or to be produced once there is what? Once there is presence of this foreign material known as antigens. So those antigens, they can be found on what? On the surface of pathogens. So this is antibody. Now, this is the structure of antibody. How is it look like? So if you can see here on this diagram, we can see this structure of antibody. Actually, it has 4 chains. 1, 2, 3, 4. Those are the 4 chains that make antibody. We have those heavy, those two, they are heavy chains. So 2 are heavy, another 2 are light. They are not heavy. So those are 2 heavy chains that consists of what? That consists of the wrong. They are wrong, actually. They are wrong chains that are heavy. So we do have also those 2 other, this one and this one. They are right chain. And then here, those 4, they are going to bind together by what you call disulfide bonds. Together, this is the disulfide bond we are talking about. So this unites this chain and this one. And then here for this, at the end of this, this chain, we do have what you call antigen binding site. That's where antigens will bind on the antibody. They have to bind here. That's why antibody is also specific. Because each antibody, it has what? It has only antigen to bind with. Are we together? So now, that binding site is where when antibody is going out to attack what? To attack this foreign material. So that foreign material has to bind with the antibody. And once they bind with antibody, either antibody is going to destroy that. It's going to kill this. Are we together? So now, this is the site we are talking about. The site where antigens can bind. That's why it is called antigen binding site. Are we together? So now, we do have this region from here to here. You see? Even here to here. Those are constant regions. They are not changing. Those are constant regions. But this for yellow color, they do change. They do change. They have what you call variable. They can vary according to the what? To the antigens that's coming to do what? To attach here. So this binding site can change in order to bind with what? With the antigens. Are we together? So this is the structure. You have now to be familiar with this structure of antibody to be able to describe it. So this is very simple. So you have this, those are long chain of antibody. They are somehow heavy. Those are two chain, this one and this one. And then we have another one, they are small. Those are two small chain. So this one, they are what? They are light, they are not heavy. And then at the end of this chain, we do have what we call antigen binding site, where antigen can bind. Are we together? So now, with those chains, they are binded together by what we call disulfide bonds. This is actually what you have to deal with, that one. So we have another one which is complement binding site. Complement binding site, where just what you call complement proteins can bind also here. Yeah, actually. We have also Hing regions. This is binding. This curve, where it is curved here, is what you call Hing region. So now, as you have seen on diagram, we can describe this in full. So is what we have talked about. Diagram consists of 4 chains we have seen, 2 light and 2 heavy, that are joined together by what? By disulfide bonds. So this light chain and heavy chain at one end together constitute what we call antigen binding site. Actually, this is what we have already seen. We have also what you call constant regions of 2 heavy chain. That region that is not changing. We do have also another one that is a variable one. So we do have now different classes of antibodies. They have to differ in their monomers and the number of disulfide bonds. Are you together? So we have different antibodies, and then they carry also different functions. We are going to see it. So now we do have different classes of antibody. We do have 5 antibodies. There are 5 types of antibodies. Number 1 is what you call EGM. This is immunoglobulin M, antibody M. We do have EGG. This is immunoglobulin G. We do have immunoglobulin A. We do have immunoglobulin E. We do have immunoglobulin D. Those are 5 types of what? Of antibodies we do have. So they have their name according to the what? To the number of the disulfide bond, and also how they are arranged, how their structure. And remember, all these types of antibodies, they are secreted by what you call B lymphocyte cells. Remember, we do have lymphocyte, which is one of white blood cells, and this B lymphocyte, it is the one that deals with what? With the production of antibodies. So those are antibodies you are having. So those antibodies, they do recognize, they have ability of recognizing millions of different what? Of different antigens. Remember, when we talk about antigens, those are the foreign material that can enter into our body and then cause what? Cause the disease, for example. That's why those antibodies we are having, they have now to do what? To bind with those antigens and then destroy them. We will see how the antibodies, they can now react with what? With antigens. So, which means those antibodies, they do offer the protection of our body. We're going to be protected because we have those soldiers. They are acting like soldiers of our body. Those are antibodies. So there are 5 classes. They are given the name according to these letters. We have M, and this IG. IG, it means this is the antibody. It is immunoglobulin M. We do have also immunoglobulin G. We do have immunoglobulin A. We do have also immunoglobulin E and D. So those are different antibodies we are talking about. So those are the structure now. Those are the structure of those antibodies we are talking about. This is immunoglobulin D. You see how it looks like? We do have also this immunoglobulin E. We do have immunoglobulin D, this one. And then we have this one, which is very complex. It is EGM. This is hemoglobin M, and then A. So antibody accounts for 8% of all antibodies. It means this immunoglobulin D, it consists of 8% of all antibodies we are having. So which means this can occupy like 8%. And then the other remaining, they do occupy only 20%, which means the high number of antibodies we are having is this ID antibodies. So this one, they are there just to recognize these viruses and what? And bacteria. So when virus and bacteria invade our body, they are going to be recognized by what? By this ED antibody. So we do have also this IgE. This is attached as what? As an individual molecule to the exposed surface of what? Of basophil. Remember, among of the types of white blood cells we do have what you call basophil. We do have also what you call master cells. Actually, those are the cells that involve in the immunity of our body. So those immunoglobulin E, it is found on the surface of basophil, just to ensure the protection of what? Of our body. We have now this ID. This ID is an individual molecule on the surface of B cells. When we talk about B cells, those are B lymphocyte cells. They are among of what? Of the white blood cells. Those cells that protect our body against what? Against the diseases. So this IgD, immunoglobulin D, it is found on the surface of B cells. Yes, they do play a role in what? In the sensitization of what? Of the B cell involved in the immunity, in the protection of what? Of our body. Are we together? Then you have this EGM. This is immunoglobulin M. Is the first class of antibody secreted after antigen is what? Is encountered. Actually, when there is antigens that present in our body, the first antibody which is going to be produced is what you call this immunoglobulin M. It is very complex. This one, it has ability of what? Of do what? Of actually, they do agglutinates. They involve in the agglutination, cramping of what? Of the red blood cells in order to prevent the spread of what? Of the germs in all parts of the body. Are we together? And then finally, we have this EGA. This is immunoglobulin A. It is found primarily in glandular secretion, such as in mucus. Actually, let me give you an example. Our saliva, this contains what you call lysozyme. This lysozyme, it acts as what? As kind of antibody that can kill the germs. Even in tears, in our tears, there is also a compound, a chemicals that act as antibody, which means it is where we can find this EGA. We can find it in the mucus, in the secretion, different secretion, including tears. That's why when there is something that's into our eyes, are we together? You have seen that the tear gland, they do secrete tears. So why tears are going to be secreted? It is because in tears, we do have some chemicals that act as like antibody, like this IgA. And those tears, they are going to be secreted in order to kill what? To kill the germs that have been entered into the what? Into our eyes. Have you seen this when there is something that enter in your eye or your eyes? There is secretion of tears. Actually, that is a kind of protection. Are we together? So those are the classes of antibodies we are talking about. Are we together? Nice. So now, we do have the function that is ensured, performed by antibodies. So what antibodies do? So number one, antibody bind to toxins produced by bacteria that cause infection like diphtheria, tetanus, effectually nullifying them. You see? Number one, antibodies, they have to bind on toxin substances that found on what? On bacteria. That are found on bacteria. If they found there, they do what? They do neutralize those toxins so that they cannot cause any problem in your body. That's first function of what? Of the antibodies. It's just to do what? To neutralize, nullifying those toxins found on what? On bacteria. Are we together? Number two, they do attach what? Antibodies. They do attach on the pathogens. And then they have now to clear them, to promote the clearness of them from what? From the body. So this is what antibody can do also. They do attach on what? On the microbial surface of bacteria. And then finally, they clean them. They clear them from the body. You see? Antibodies are very serious, my friends. We have this kind of protection. If you don't have antibodies, do you think what's going to happen? We can stay in our life every time we get sick. But because antibodies are there, we are somehow protected. Number three, antibodies form covering on bacteria and virus. They cover them. So they not allowing them to gain entry into the tissues. You see? Antibodies, they do cover. They make a cover on the virus and what? And bacteria. So that they cannot get into our tissues. If they are covered, they are not allowed now to get into the body. You see how antibodies are working? Nice. Number five, antibody, like this immunoglobulin G type, is highly mobile, capable of leaving saturation and reaching the skin where it can neutralize the surface of bacteria. You see, we do have some bacteria that are mobile. They can move. They can move from through the blood, then reaches on the skin where there is what? There is bacteria, where there is germs, where there is virus. And then finally, they go there to do what? To neutralize those bacteria. Once bacteria are neutralized, they cannot cause us the disease. Are you together? So we do have also antibody, which is immunoglobulin A, found in large amount in mother's milk. And then help protect the newborn against infection during the first month of their life. We have this antibody, like this antibody A, immunoglobulin A, which is found in the breast milk of the what? Of the mother. Once the newborn, the baby, suck the mother's breast milk, the baby can get what? Can get that antibody A, immunoglobulin A. And once baby gets those antibody, they can protect them through their life. Are you together? So now, that's why it is very necessary, it is very important for the young ones, for the babies, to suck their mother's breast milk. To get the breast milk from their mothers, it is very essential, because the baby can get some antibodies from there. Are you together? So finally, we have this immunoglobulin E, which is playing important role against parasitic infection. Through, it is also responsible for allergic reaction. So now, we do have also this antibody, which is responding to the what? To the parasitic infection, parasite. When we get a different parasite in our body, sometimes there is allergy. There is allergy which is going to be produced. So this antibody E, it is the one that involved in this allergic response to various agents in the environment. So the next lesson is just we describe about this allergy. Are we together? So when the allergy comes out, it is kind of what? Of showing that there is now clearance, there is neutralization of what? Of these germs from the body. Are you together? Now, we do have the reaction between antibody and antigens. So how antibody destroy microbes? How is it going to carry out that one? Number one is by neutralization, to neutralize. Once the germs, once the virus, bacteria, and so on, are present. So what the antibody do, they are going to react with the antigens of what? Of those germs. And then finally, they cover them, then neutralize them from working. They do not work. That's number one, neutralization. Number two, it is about agglutination, where the binding sites of the antibody molecules attached to antigens on different microbes, thus causing microbes to do what? To clamp together. So they do clamp together. They are going now to be together. In order, they can now be easily destroyed. This is what you call agglutination, which is going to be ensured by what? By antibodies. Number three, another mechanism of now destroying what? Antigens of what? Of the germs is by what you call opsonization. Opsonization. So what's happened? So the molecules are going to coat the surface of antigens of what? Of the germs. Once they are coated, this one, it will be easy for what? For the white blood cells to do what? To engulf them. This is what you call opsonization. Another way of antibodies to do what to do, to react with the antigens is just do what you call precipitation. The antigens, they have now to be soluble. They are going to be soluble into the antibody molecules. Once they are dissolved into antibody molecules, this one, it can help us to be destroyed easily. Are you together? Then lastly is lysis. When we talk about lysis, it's like destroy. Once the antibody react with the antigens, or those are the foreign cells, foreign material, they are going now to do what? To be destroyed, to be busted. The cells is going to be busted. Either the germs, pathogens, they are going now to be busted. Once they are busted, they are going now to be released outside. Are we together? Nice. This is what we are talking about. So those are actually what we have already discussed about. This is neutralization. You see? Those antigens have been covered. You see? They have been covered by antibody. Then here it is agglutination. They form a clamp. They are binded together. So to release them, it is also, this is precipitation. You see, they are dissolved. They are dissolved into antibody surface. And then finally, we have these rises. You see? So those are the mode of action of antibodies. Before I give you the homework, we can try to understand what is antibody. When we talk about antibody, what do you understand about that? Nice. That is a protein that fights against what? Against pathogens in our body. You are right. And then how antibodies, they do what? They do react with the antigens. Number one, neutralization, you are right. Number two, by neutralizing the antigens. Number two is by agglutination. What is agglutination? Is when the antigens, they are clamped together. So once they are bounded together, it is easily just to destroy them. Number three, it is by precipitation. Are you together? Where antigens are going to be dissolved into, they are going to be dissolved into antibodies on the antibody surface. Then also we do have these rises, where antigens for the germs they have now to do what? They have to be destroyed. They are going to burst, and then they are going to display out of the body. Are we together? So we have seen different classes of what? Of antibody. Do you remember? Number one, we have EGM. EGM. EG means immunoglobulin M. We do have immunoglobulin E. We do have immunoglobulin A. We do have also immunoglobulin B. And then finally, we do have B. Are we together? So you have time to do what? To make your revision. I hope you can continue to read, and then you will have more details about this. Are we together? So now, so we do have now this homework. So describe the mechanism by which antibody destroy extracellular microbes and antigens. So you explain the mechanism how antibody can destroy extracellular microbes and what? And antigens. So I hope you will continue to enjoy. I can say goodbye. We shall meet by next time. Thank you.